Recording in progress.
Good morning, everybody. Feels like Monday. Let's get a little bit more brightness. Okay, so welcome to my office. One cool thing about my office, it has an enormous whiteboard. And it has a coffee machine as well, which is also really neat. So I'm never short of coffee. Right, so today we need to quickly go through that intro PowerPoint and the flow of genetic information PowerPoint. That's kind of what's on the list for today. Before we start that though, ooh, Jess, I know exactly where you are. That's a lovely spot. I wish I was out there. That'd be much more comfortable. Yeah, but then I get like distracted by watching like birds and bees and things. Lizards as well. There's some really cool lizards that live out there. Anyway, before we get going on any of that, does anybody have any questions from Thursday about assignments, course structure, you know, any of the jazz that we covered on that day? Probably not, but if you do, that's super cool. I'm just kind of trying to get myself into the habit of giving people time to ask those questions, right? So we have that kind of space at the beginning of each class period to go over previous stuff. You know, that whole make your list of questions kind of deal from going over your notes. But as we haven't got any information, then, you know, you probably don't have any questions. Okay, so get up chat and share the screen. I actually had I can't remember who it was, must have been from my other section. Someone give me a tip about managing all of the tabs that's open in my browser, which I thought was pretty funny. I know where everyone is. I've got my own system, believe it or not. It just gives people conniptions whenever they see it for some reason. Okay, so essentially what I want to do today is give you a general overview, you know, of what's what. A little bit of history, not an awful lot, because, you know, science is generally forward-looking rather than backward-looking. But also in the second PowerPoint, which we'll spend more time on, going over some of the basics, right, particularly of molecular biology, right? So kind of really just making sure everybody's up to speed, right? So that you're kind of as much as possible on the same page, right? Because I know that there's a big variance between students in terms of how recently you took GenBio 1. You know, actually how recently you've been in college. Some of you, you know, you're coming back after maybe 10, 15 years of pursuing another career. Others probably did GenBio 1 a couple of semesters ago, right? So there's a big variation. And I want to make sure that I don't leave people behind. Okay, so in terms of what we mean by genetics, right? So there's really two kind of aspects to genetics. I've got this cool chair which lets me like, so try not to make you dizzy. One is what I like to call the nuts and bolts, right? I like working with my hands. I'm very much a mechanic machinist type person. And really what I mean by that is like, how do things work, right, at a genetic level? And you always have to start with DNA. All living organisms, and I don't include viruses in that, but all living organisms use DNA as a store of information, right? So what does DNA look like? What are its properties, right? What are its dynamics? Because DNA, even though it doesn't go anywhere, right, it stays in the nucleus at all times, it is dynamic. And we'll spend pretty reasonable bunch of time later on in the course talking about how DNA changes, not its sequence. That always stays the same unless mutations occur. But there are many other ways in which DNA can change its accessibility and so on. Right. What comes after that? Okay, so really then we're talking about transcription and then translation, right? How does that work? How do changes in DNA affect the process of gene expression, or what is produced by gene expression? You know, or where, or how much, right? So there's a lot of kind of how do things work? You know, really in many ways, this is like opening up the hood of a car and kind of poking around in the engine bay and going, oh, that's connected to this and that does that, right? That's in many ways what molecular genetics is really all about. And how does that vary both within taxa, right? So in other words, why are we largely genetically identical, right? And, you know, we're not identical because we're not clone troopers, right? This isn't Star Wars. But if you look at the difference genetically between all of us, even though we come from many different places genetically, right? I'm a Northern European, I guess, German, Scottish. That explains many things. And also various other bits and pieces, right? Probably some rampaging Vikings and Saxons and stuff at some point, right? But despite that, we are largely identical, right? We are more similar than we are different. And yet clearly we are very different, right? So how does that work, right? How can we be so similar to chimpanzees, right? Which is our kind of closest evolutionary branch, I guess one could put it. And yet we're clearly really different from chimpanzees in many ways. Some ways not so much, right? So how does all that work, right? What's the stuff underneath, right? If you kind of peel up the you know, like someone's skin, like, or surface of an organism, like what's going on underneath? But that's in an individual, really, when you think about it, right? Why am I the way I am? Why are you the way you are? But we're not individuals in isolation, right? We're part of a lineage, essentially, right? And so we are the way we are because of our parents. Not entirely, right? There is a degree of nurture going on, not just nature, right? But we are, all of our genetic material comes from one or both of our parents. I'll get to that a little bit later. It's kind of a funky idea, right? So why are we expressing certain phenotypes and not others, right? How do those traits, such as hair color, height, propensity to cardiovascular disease, right? How do those traits get inherited down through the generations, right? Why is it you can look at, you know, your family history and go, ooh, there's the crook nose, dun, 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 dun. Or the flip side, why is it that, you know, both my grandfathers were really quite tall for their age? They kind of shrunk a little bit by the time I got to know them. But, and my brother and I are pretty tall. We're both over 6 foot. And both my parents are like, you know, not so tall. I mean, I'm not saying they're small, but they're not particularly tall, right? My dad always says it's because of the milkman, but it could also be because that trait, you know, kind of disappears, right? Or that phenotype disappears and then reemerges, right? And so that's really the study of inheritance, right? A generational issue. Now we're not just talking about individuals in and of themselves. Now we're talking about individuals as a lineage, right? What did you get from your parents? What did you give to your offspring? So on and so forth. And so really, and this is again part of the kind of vocabulary deal, right, which I talked a bunch about, I think, on Thursday, right? You're going to learn the language of genetics. Oh, well, that's nice. Thank you. Just telling me that you can now see my screen. So us being humans, we like, slicing and dicing things up quite neatly. It's just what we like doing. I'm sure our ancestors organized rocks into nice piles of equal shapes or sizes. We like to subdivide things, and these are very much really artificial divisions. They're just useful for understanding stuff. So really starting off, actually, I need to change this over. Let's move that up there. Kind of makes more sense that way. There you go. Sorry if that screws up anyone's notes. So really at the smallest scale, right, you're talking about molecular genetics, right, the nuts and bolts deal. When you go up in scale, and that can be at, as I said, the individual level, but when you start going up in scale to many individuals and inheritance, now you're really talking about transmission genetics, right? How are those traits passed from one generation to the next? What's the probability that your offspring will express a particular phenotype? Right, so a lot of that is kind of math and logic based, right? That's something that we'll be probably dealing with first. Well, we will be dealing with first in this course, right? Kind of in many ways, people call that Mendelian genetics. That's because Mendel pioneered that particular approach. And then when you go up bigger in scale, right? Now you start looking at populations and populations over time, right? And time is really one of the critical factors in evolutionary genetics, right? Because really you're going to be starting to look at population scale genetics, what alleles are present, so what versions of genes are present in that population, and how does that frequency change over time? You can actually, San Antonio is an excellent example, right? It's clearly based in central Texas. There's a long history of migration in this kind of part of the world, Mexico, Texas, maybe a little bit further along the coast, but not a great deal. It's mostly between those two parts of the world. And now you have people coming in. There's always people moving in, like ourselves, right? My family from outside of Texas, right? So we're bringing in alleles which may not have been present in that population, right? So the genetic makeup of San Antonio is changing over time. And it's not really in a good or bad way, because there's no kind of judgment associated with that. It just is changing because there's gene flow, migration, people are moving in and out. We're not really going to deal with any of that in this course, because that is dealt with very thoroughly in evolution, which is kind of a follow-on course from genetics, much the same as GenBio 2 is for GenBio 1. So that you'll take either next semester or the semester after that. But these are very artificial divisions. And really, if you're studying genetics, almost certainly you're going to study, your work is going to blend into different areas, right? So I'm very much a molecular geneticist. I study, how genetic pathways work, but I also use transmission genetics and the principles to do my research, right? If I want to get two different alleles together, I need to know how many worms, because I work on C. elegans, it's a small nematode, do I need to look at to be able to be assured of getting the combination of alleles that I want, right? So again, it's just another example of how these things aren't real divisions, really. They're just ones that we give genetics. Genetics very young. So really, you could argue that genetics as a modern science took off with the publication of the Human Genome, which was in 2001, I think, 2002. Actually, here's a fun question. Anybody know in current dollars, I guess, how much it costs to sequence the human genome? One assembled genome sequence. So all of the DNA from someone, I don't know who got their genome sequence first. Anybody know how much that cost?
It's about 150 million, I believe.
Maybe back then. Does anybody know what it costs now? Well, it would have cost if it was done now. $1 billion in modern money. Isn't that crazy? And it took hundreds and hundreds of people. I think it was about 3 to 5 years. I can't remember the details to do it. It's an enormous enterprise across the world between largely the US and the UK, but also other countries. Do you know how much it costs to get your genome sequence now? Like if you wanted to, because you wanted to know.
Like $100.
A little bit more than that, about $2,000, give or take. Right, and so that allows you to do a lot of really, really cool stuff, right? So if you get a particular disease, cancer especially, you can just get your genome sequenced, figure out what kind of cancer molecularly do you have? Are you resistant to particular treatments or not? Things like that. And so, genetics has really taken off in the last 20 years. That was kind of kicked off by the discovery or, I don't know, development of something called PCR, which stands for polymerase chain reaction. There's a T in there. And only one N, sorry. I'll edit that before posting that up, maybe, if I remember. Right, which was really in the kind of mid-70s, I guess, really came into its own in the early 80s, right? That allowed us to copy DNA, to amplify it, to clone it, to stick it somewhere else, right? Anyone that's diabetic most likely owes their life in some degree or other to that, because insulin was the very first gene ever cloned and expressed. Right, which meant that we didn't have to rely on pigs and their pancreases to provide the insulin for diabetics to manage their blood sugar levels, right, which is pretty cool. And then it goes back a bit further to, I don't know, probably Watson and Crick and Franklin and what was his name? Wilkins? Yeah, there you go, Wilkins. These two dudes and a couple of others who elucidate the structure of DNA, right? And so that was in the early 50s, right? So really, it's not very long ago, 70 years at most, that we've gone from not knowing what the structure of DNA is to being able to determine to a base, like to a letter, what an individual's genome sequence is, right? And what that means as well, more importantly, which I think is just absolutely nuts. So it's a very fast-paced science. And as you can tell from, the development of CRISPR-Cas9 genome editing, which was really in the last 10 years at most, which gives us the power to genetically edit organisms, including ourselves. Kind of makes you wonder what we'll know and be doing in 10, 15, 20 years' time. It's a very exciting field to be in. Okay, so going to do a little bit of recap, and then we're going to move on to the other PowerPoint. Might finish a little early today. We'll just have to see. Oh, I've also discovered GIFs or emojis, sorry. How cool is that? Someone did a rubbing chin emoji last class, which I thought was really cool. Like, huh. Anyway, I also just got my first smartphone, which I keep telling everybody. And so I spend most of my time annoying my children with GIFs. That's my annoying them from a distance, which I think is super cool. I know it's taken me a long, long time. Still not quite used to it, but I'll manage. Yeah, I actually had one of those grandpa flip phones, you know, the ones with the really, really big numbers. But it was a complete turd. Absolutely hated it because it was junk. But anyway, people always said that I sounded like I was talking from the bottom of a tin can. So a lot of this stuff is going to be, you know, stuff you already know, which is great. For some of you, it might be a useful recap. And I'm going to start kind of putting what my grandmother would call worms in your ear. Right. And so many things that we cover in this course will be introduced in some form or other ahead of time. And so what I try and do is say, okay, this thing that we're learning now, well, we're going to not just need to know it, but we're going to use that later on, right? So I kind of try and put those worms in your ear now so that when you do hit it, you're like, oh, right, yeah, awesome. Okay, so first up, structure of DNA. DNA is a very stable molecule. This is where I like showing everybody, get out of it. How's his name spelled? Ötzi the Iceman, which is super, super neat. This guy, he was spat out of a glacier in Austria after lying there for what they think about 4,000 years in the Austrian Alps. And they think, where's the nice animation? There you go. They think he was actually shot in the back with an arrow, which I think is terribly rude. I don't know why. No one knows why, right? But they found kind of a nick in one of his ribs, I think, which is similar to what one would expect with an arrow or an arrowhead. And they were also even able to figure out what he had for his last meal by sequencing the contents of his stomach. Right? This is 4,000 years old. He's clearly not in particularly good shape, right? He's a bit, you know, shrunken and wizened, fairly well preserved. But DNA is super robust, right? You can even, you know, sequence DNA isolated from bone marrow of our distant ancestors, I guess one would call it, from tens of thousands, if not hundreds of thousands of years ago. Right? That's how we kind of can figure out in one way what our evolutionary tree looks like. You know, Australopithecus and Homo erectus and things like that. And part of the reason for that robustness, let's get back to where I was, is this double-stranded structure. And so the kind of reactive, vulnerable parts of DNA are on the inside. That's the nitrogenous bases, these rings that you see here. And so the outside, which is called the, let me just blow up this a little bit, called the phosphate sugar backbone, right? So it's P stands for phosphate. And then S stands for sugar. Right. you'll see that is an alternating pattern, right? So phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, right? And that's because one nucleotide consists of a phosphate, a sugar, and a nitrogenous base. And that nucleotide is often called a base, which can be a little confusing. And so the phosphate and the sugar is the same for every, well, the 4 different DNA nucleotides. And it's the same for the 4 different RNA nucleotides too, even though they differ between DNA and RNA. It's the nitrogenous base which differs. Where's the chat? There we go. There are 4 different bases, right? Adenine, thymine, guanine, and cytosine. Get to those in a second. And another thing which is really, really important, and we'll come back to this a bunch in the future, is DNA is not parallel. It's anti-parallel. So you have one strand going in one direction. You have another, the opposite or paired strand goes in the opposite direction. And so you can see that here you have 5' end, and we'll get to what that means in a while. We have a 5' end, and down here you have a 3' end, right? So DNA has direction, right? It's not like a polysaccharide or something like that, that can be, you can't tell one end from the other. But if you look at the other strand, it is the opposite. So the bottom is the 5' end, and at the top is the 3' end. This is really important for DNA replication and for transcription. Okay, here's a question for the crowd. What holds these bases together? What holds the 2 strands of DNA together?
Hydrogen bonds.
Yeah, exactly. Hydrogen bonds. And does that differ, or do they differ in number between the different pairs? So A pairs with T. Yeah, that's right, Clarissa, but how? How do those, yeah, also, thank you, Rachel. So there are fewer bonds holding adenines and thymines together than guanines and cytosines, right? 2 versus 3. This is also really important when it comes to opening DNA up. So regions where you need to open up DNA, what would you expect to find more of? A's and T's or G's and C's? Right, A's and T's. Also, the other way in which they pair, and this pairing is really important for DNA replication specifically, but also transcription as well, which is a little bit different, is that you notice that there's this unequal pairing, right? So we have a 1 ring always bound to a 2 ring and vice versa, right? And so you only ever have a 1 ring base, which is called a pyrimidine. paired with a 2-ring base, and that's a purine. Right. So cytosine, let me just put that big. Dang it. Quit that. Autocorrect is a real pain in the bum sometimes. So cytosine and thymines are pyrimidines. Adenine and guanine are purines. It's not a crazy important piece of information to know, but it's the size deal and the hydrogen bonds together are how you get this complementary base pairing, right? So those two things together are what make that AT and GC pairing. Very specific. It's a big part of how DNA replication is so accurate, which is really important because you don't want to have mutations popping up every time your cells divide. It would be a really big deal. Okay, anybody got questions so far? Time to catch up on your notes.
What does the 5-end and 3-end mean? I forgot.
Oh, good question, Jesus. Is it Jesus or Jesus? Which do you prefer?
Jesus.
Jesus. Okay. So this refers to which carbon on the sugar the thing is attached to. Right. So actually, do we have a better view of this? We do. Kind of jump ahead a little bit. Come on. There we go. That's a better view. So the 5-ring ribose sugar, right, which is part of the backbone, you count the carbons clockwise from the oxygen. So the first carbon is here. You can't see it because, you know, we don't put all of the atoms into these models. The first carbon is where the nitrogenous base is attached. Pardon me. Second carbon doesn't have anything in DNA. It does in RNA, but we'll get to that later. And the third carbon, actually, if you go all the way down here, has a hydroxyl group. And so that hydroxyl group is essential for adding another nucleotide onto it. So DNA can only get added or made in the 3' direction. You can't add anything to the other end, right? So that's why DNA replication is directional. Keep going round and you see the 5th carbon up here that has the phosphate group attached to it. So if you have linear DNA at one end of it, you'll have a phosphate hanging in the breeze, right? That's why this is called the 5' end and this is called the 3' end. It's basically a shorthand telling us, okay, at this end we have a phosphate, at this end we have a hydroxyl group that is available, one presumes, for adding another base onto.
And you said you can only DNA replicate on the 3' end?
Sorry, Keanu, are you asking about nucleotides can only be added on the 3' end?
Yeah.
Yes, that's exactly right. And that's the same for RNA too. RNA and DNA can only be synthesized in one direction per strand, at least. Now, if you think about it, and here's another worm for your ears, DNA is anti-parallel. So you have one strand going down like so, and one strand going up. So when it comes to replicating your DNA, you're going to have DNA replication going in a divergent direction. But we can't have that happen because we have to make sure all the DNA is synthesized at the same time. So that causes other problems which have to be solved. right? So this is why that's why I like spending time on this intro stuff, because a lot of the kind of more complex things that we'll be studying later on are founded on this, what we're learning now. So yeah, good questions. Thank you, Jesus. Any other questions? All right. So obviously, once you know the structure of DNA, you can start figuring out how it's copied. You can figure out how it contains the information that's used to make proteins or RNAs, depending on what we're talking about. You can figure out how changes in that information change the expression of that protein and its function, right? That's really the mutations are the basis of all novelty, whether good, bad, or indifferent, right? That's the only way, yeah, mostly the only way in which you can change. It's a real pain sometimes teaching this course because I have to add so many caveats, because DNA sequence change isn't the only way in which your DNA can change, but we'll get to that later. And last of all, we won't be spending a lot of time on genomics. Actually, I don't think we'll be spending any time in this class. You'll cover that later on. But it's really worth introducing the fact that really genes do not act in isolation, right? Or proteins don't act in isolation. And there's a lot more to you as an individual than just your protein coding sequences, right? Just the genes that produce proteins. There's a lot of other information which we really don't actually fully understand, right? That's one of the cool things about teaching genetics is it's very easy to say, I don't know, right? Because there's a lot of stuff that we don't know. And there's a lot of stuff, if I was teaching this 10 years ago, known even less, right? And so genomics is really kind of studying the whole picture in terms of your genetic information, right? Oh, and one cool fun fact. So how many genomes do you have? It's a little bit of a tricky question. How many genomes do you have as an individual? You're probably thinking, what's the trickiness there? That would be the straightforward answer, Clarissa, but it's not quite right. Oh, those are chromosomes. That's kind of right as well. Chromosomes are the, I guess, the books, right? Where your genome is the library or the shelf, right? So you actually have two genomes. One nuclear genome. So that's the DNA in your nucleus. And you also have what other organelle used to be, we think, a bacterium, do you have in your cells? Almost all of your cells, some more than others. If I start saying it's the powerhouse.
Mitochondria.
Yeah, I always call the powerhouse of the cell. Right. And so you have a mitochondrial genome. And that comes from your mother only. Right, so you have DNA in you that did not come from your father, which is kind of neat. So you can actually trace your genetic lineage back potentially thousands of years in a matrilineal way. So I got my mitochondrial DNA from my mother, she got hers from her mother, she got hers from her mother, and her mother, and her mother, and so on and so forth. It's actually pretty neat. Not many genes, it's very small, 20 maybe, 19, something like that. But still, that just comes from your mother. Anyway. All right, I'm gonna switch PowerPoints. Go over here. Anybody have any questions before moving on? Okay, let's stick that down over there. I gotta say, I have always had so many fights with the chat box. All right, I'll just leave you there then. Fine. Sorry, so if I'm look like I'm, you know, surfing the net or something, it's not. I'm fine with the chat box because it's always bloody in the way. Okay, there we go. All right, so this PowerPoint is really all about taking you from the basic structure of DNA, right? We'll be getting into the more detailed structure later on, particularly when we talk about chromatin. And then we're going to go through transcription, translation, right? So the basics of gene expression. Again, we'll be covering these in more detail. This is really just to get everybody up to speed kind of deal. And then we'll talk a little bit about kind of mutations and stuff like that, which is a kind of fun thing to talk about. And so we've already covered this in large part, but one thing to note as well is that between these nucleotides, and it's really just here or around there, the nucleotides, the individual nucleotides are linked together by what are called phosphodiester bonds. Phospho because there's a phosphate there, ester because that's a particular type of bond between a carbon and an oxygen. Right, so these bonds are created by the condensation dehydration reaction between an incoming nucleotide and this hydroxyl group, which generates water. So you have to have that hydroxyl group there for another DNA nucleotide to be added to it. When that water is removed by DNA polymerase, you end up with your phosphodiester bond. DNA also has we've already covered this in large part, a neat, what I like to call topography, that kind of shape, essentially, right? It is not an even twist. So if you take a twist tie, for example, off a loaf of bread, twist it together, most likely you'll get a fairly even twist, or braiding hair, you know, that kind of stuff. Obviously, you can't do mine. Always been tempted to grow my beard out so I can do like the Viking thing with beads and stuff. But anyway, my wife doesn't let me. DNA is not an even twist, right? It has what's called the minor groove, which you can see here. It's a small groove, and the major groove. And these are important because proteins actually recognize this shape. So proteins not only recognize the sequence, which you can see here, like the individual bases or the information contained within them, they also recognize the particular shape of DNA, right? And so I think the tryptophan repressor from memory actually binds into 2 adjacent major grooves. So the shape of DNA is also important. Okay, so one thing I like to stress in my classes is that DNA it's not static, right? But it doesn't do anything, right? It doesn't have a function other than being the store of information and being the means by which that information is passed from one generation to the next. Right, so it's very much like a library, right? And, you know, inheriting a library from your parents if they have a particularly cool one and they pass away. Other than that, it does nothing, right? A cookbook cannot bake a cake. It'd be kind of cool if it could, but it can't. Right? You have to use the information within to be able to create the thing that you want. And that's really what DNA is all about. So even though it contains the information needed for a particular phenotype, it doesn't create that phenotype itself. It has to do that via, well, it is done, not that DNA does anything, doesn't have to do anything, right? The phenotypes that you see, or even ones you can't see, are mediated by RNA and protein. And the process by which those are produced is called gene expression. And gene expression is really, I mean, this is called the central dogma. It really should be called the sequence hypothesis. It's a bit of a misconception. One of my bugbears of teaching genetics. I'll have to change that at some point. Yeah, actually, let's do it now before I forget. There you go. I'll upload a new version later. And comments, notes. Basically, a sequence hypothesis, and this is from Watson and Crick, is DNA, mRNA, protein. Central dogma, which is funny because they actually expressed regret for using the word dogma because it caused them all kinds of grief afterwards. I don't know how to do the basically, a central dogma is you can't go backwards. That's what the central dogma means. We actually know that that's not 100% true. It was a thought exercise. We can actually go from RNA to DNA, but we'll worry about that later. Anyway, my little bit of pedantry aside, and it's important, you know, I think. Basically, it's going from DNA to protein, right? How do you get from DNA and the information that it stores to something that can do something? Whether it's an RNA, some RNAs are active in one way or another, but most of the time we're referring to proteins, right? But how do you get from information here to here? And that's really the culmination of a whole bunch of things going on, but we can simplify it to transcription and translation. Transcription is the creation of RNA, whether it's mRNA from the gene being expressed or ribosomal or tRNA, transfer RNA, which are important in translation, which are also expressed from genes, obviously. All of those together, along with a whole bunch of proteins that create the ribosome, translates that mRNA into protein. That whole process is called gene expression. And that's typically how I'll be referring to it. I don't generally call it the sequence hypothesis because no one knows what I'm talking about. So transcription, I love this this diagram. This is super, super neat. Let's see if I can make it bigger. So this is actually a super cool diagram because there's a lot of information in here, right? One is that to be able to transcribe DNA, you need to separate the two strands. So you use one strand as a template. And you make the RNA complementary to that, well, to that template. I have to put my hands like so. Still anti-parallel, right? So you can see here's the DNA strand in kind of grayish blue. 3' down here, the RNA transcript is 5' to 3', right? It's anti-parallel. And it's got the little arrow at the end because that's the direction in which it's being made. Also, you can see in here that RNA has a different base instead of thymine. So normally in DNA, it would be T and A together, or A and T. In RNA, it's going to be U or uracil, the base pairs with adenine. Apologies, I don't actually have like really huge hands. It's just my camera on this laptop is down here rather than up there. That also is why I look like I'm super reclined in my chair. So this is a really cool image because it shows an awful lot of what's actually going on. It's one of my faves. And so in terms of transcription, we've already covered the bottom two, right? One strand of DNA is used as a template. This is not the DNA strand which contains the information in the gene, right? Because we're making a almost direct copy of that strand in mRNA. So if you look over here, right, where we have a T in our coding strand, we have a uracil. Where we have an A, we have an A. Can't tell you what that one is, but where we have a T, we have another uracil, and so on, right? So this mRNA strand is a direct copy of the coding strand, but it uses the other strand as the template. And the reason for that is that if you want to copy this in a 5' to 3' direction, you can't use this as the template because you'd need to make the DNA in the opposite, the RNA in the opposite direction. So you have to use the other strand to get it going in the right direction. Also, this whole process is controlled to varying extents by regulatory sequences, right, which are recognized by proteins. And those are what determine when, where, and how much a gene is expressed. And this also just reiterates that uracil is the base in RNA. Also, another key point too which will, I think we cover this later as well. So this is a DNA molecule. If this were RNA, instead of an H here on the second prime carbon, you would have an OH. So RNA nucleotides well, that's also called a hydroxyl group. That's why RNA is ribonucleic acid. DNA is deoxyribonucleic acid. Ah, bugger you. Stop doing that. Because that extra OH has been removed. Hence deoxy. That's why it's DNA. Okay, so essentially we now need to translate. So RNA is a copy, it's a transcript. It's no different to the transcript that you get from the registrar's office, which is a copy of all of your grades and the courses that you've taken and things like that, right? And so the purpose for mRNA is to get the information out of the nucleus into the cytoplasm, at least in eukaryotes, in a form which can be translated into protein. Right? You can't translate DNA into protein. It doesn't work that way. And so what we need to do is we need to literally translate the information in the mRNA into an amino acid sequence. Which essentially is a protein, right? Once a whole bunch of other stuff's been done to it. And so the way in which we do that is we use these translators, essentially. These are RNA molecules called tRNAs, right? Short for transfer. They're basically the translator. And they turn 3-letter mRNA words into an amino acid. And they do so by recognizing those words, essentially. Right, so again, this whole deal of base pair complementarity, right? Particular bases sticking to a very specific other base. You see that exactly here. So you have an AUG, that's a word, right? That's actually a words in mRNA are called codons, right? So you have the AUG codon, that's the start codon. The tRNA that recognizes it has an anticodon, which is the opposite. And so you'll see here you've got adenine base pairing with uracil, uracil with adenine, guanine with cytosine, right? And at the other end of that tRNA, the amino is the amino acid. Right, so these are, I actually like calling these the Rosetta Stone. If anybody knows what that is, it's a series of tablets found in I think it's by the French army for some reason, in Eastern Mediterranean, which had things like Sumerian and stuff like that, but also hieroglyphics. It's a series of instructions for something or other. Anyway, that's what allowed hieroglyphics to be translated, which is pretty cool. A little bit of history there. Oops, gone on too far. And so this sequence of words is encoded in DNA. That's the information. That sequence of words is basically read by the ribosome with the aid of these tRNAs, and that's used to create this sequence of amino acids, which is the protein. Now, obviously, there's a lot more to it than that, but really that's the basics of it. That also gives us a genetic code, right? And so because we're reading these in words of 3, we have 64 codons and way more codons than we need for the number of amino acids that we have. Right, going to point out a few little things, which we'll again come back to later. We have this one here. Thionine is our start codon. It's at the beginning of every protein, bacterial, archaeal, eukaryotic, whatever, at least at the beginning of translation. We also have 3 codons which are not recognized which don't have a tRNA. These are used to stop translation, because obviously you got to stop it at some point, right? At the end of whatever protein sequence is. These are called stop codons, right? I'll come back to that later. And so all of that basically ends up creating a protein. Now, one thing to point out, people often think of RNA as a very simple molecule. Actually, it's far more interesting a molecule than DNA in many ways. And obviously, I'll probably get in a fight with someone who studies DNA if I said that. But RNA is really, really neat. So it can bond with itself, right? It can create folds, it can create dynamic folds as well, which change. And also RNA can act as an enzyme. So it can both contain information.
Are the folds there for any specific reason?
Oh, most certainly, yes. The shape of RNA is very, very important to its function, just the same as the shape of protein is. And so, yeah, so essentially, I'm kind of get to in a moment in the chat, but this particular shape creates a catalytic pocket that allows it to catalyze a reaction. And so it has to form this particular shape, which is a bunch of loops and stems, and they look like lollipops, basically. Let's see if I can find. So if you're looking at it kind of simply, like in a 2D fashion, this is what tRNA looks like. I don't actually know what these other arms do, but this one is involved in binding to mRNA. If you look at it in a 3-dimensional model, it looks very different. Actually, I think these might be part of how the amino acid gets added to the end up here. I'm not 100% sure though. But yeah, the shape that RNA forms is really important. And actually we'll be looking at that. Bacteria use that as a means to control gene expression. It can do, yes. And so again, that's something that we'll look at in quite a lot of detail for control of gene expression in bacteria and prokaryotes. So depending on which, sometimes it can only form one shape. but other times it can form more than one shape depending on which bit is free to pair up or not. So it depends basically on the type. But also RNA can contain both information, which is like DNA. Good questions though, Kiana. And catalyze reactions like protein. And that's called a ribozyme. And so the classic example of a ribozyme is, where are we at? Ribosomal RNA. So ribosomal RNA is the molecule that synthesizes proteins. I mean, it does it in a ribosome, but it's not the protein part of the ribosome that makes protein. It's the RNA part, which is pretty wild. And just for a complete whoosh, tangent, hence the RNA world hypothesis. And that basically posits that life started as RNA, because it's the only thing that can both contain information and catalyze reactions, potentially making itself. So that's just something to think about. It's not the only hypothesis as to how life started, but it's a pretty cool one. Anybody got questions? Yeah, RNA is really cool. Okay, just coming to the end now, so you'll be free to let your brains cool down in a few minutes. So mutation or series of mutations, chance mutations too, right? So mutation is random. All mutations naturally occurring, i.e. ones that we don't create in the lab, are completely random as to where they occur, right? And those mutations are the only really source of novelty, right? So you or us as a species are the way we are because of the result of potentially millions of mutations over hundreds of thousands of years and selective pressure from our environment in various forms that has selected for particular mutations and made them part of our genetic background, right? So really, you know, there's no evolution without mutation. It's actually, sorry, going off on another tangent, it's actually a really, really cool way of thinking about how life evolved, right? Because I teach astrobiology once in a blue moon, or even rarer, right? And it's one way in which Earth is like this Goldilocks kind of environment, because we do have radiation that causes mutations, right? UV radiation from the sun, gamma radiation from the sun, and cosmic radiation. But we also have this awesome magnetic field around us, which causes northern and southern lights, which protects us from most of that, right? So if we were, say, around Proxima Centauri, right, which is a red dwarf, they produce huge flares of radiation kind of randomly. There's a good chance that we wouldn't have evolved because we would have had too much mutation. You know, other potential exoplanets, there might be not enough radiation to create mutations, which are the basis upon which natural selection acts. So it's a really cool thing to think about in terms of, you know, how life occurred on Earth and potentially not anywhere else that we know of yet. Anyway, most mutations are neutral, like they don't change anything, and we'll talk about why later on. Quite often they're deleterious, right? So they're bad for you because they stop something working, right? And examples of that would be, things like sickle cell anemia, let's say, or cystic fibrosis, right? Where proteins don't have the right structure or function. And we can classify those mutations in a whole bunch of different ways, right? So this is not something I'm going to expect you to, I'm not going to quiz you on like what all of these different things mean, right? Because it's not super important, but there's like really fun stuff in here. So for example, where those mutations occur matters a great deal. So if I had, or this, I'm not having any more kids, but let's say before I had kids, I, took my pasty white British self out into the sunshine and got sunburned, right? And so mutations occurred in my skin. Would any of my future children inherit those mutations? Yes or no? If those mutations occurred in my skin or something like that. No. Why, Maria? Or Maricela? Either of you. Why would my children not inherit those mutations? Whatever they might be. Oh, the DNA is most definitely being compromised because they're mutations. And there is genetic for sure. That's right, Louise. It's because it only occurred in the somatic tissue. Somatic tissue is basically anything that isn't involved in creating the next generation. Right. So essentially, the only way in which mutations can pass on to my children, or could have before I had children, were if those mutations were present in my germline, in the sperm, essentially, or if you're a female, in your oocytes or your eggs. Right. So where those mutations happen matter if we're thinking about inheritance and evolution. Obviously, if those mutations occurring in my skin and I end up getting skin cancer, it matters a great deal to me, right? But that mutation ends with me, right? It doesn't get passed on into future generations, right? What kinds of mutation you get also makes a big difference, right? If there's a mutation, let's say, let's go up here, in a coding region, right, which turns our CUU codon for leucine into CUA, is there going to be any change to the protein? No, there's not, because it still encodes a leucine, right? So this is called a silent or synonymous mutation, right? If it ends up instead creating a stop codon, then that's really bad news because you're going to get a truncated protein, which probably isn't going to work, right? So what kind of mutation you have often has a really big effect on the effect of that mutation or the consequences of it. And in turn, if you look at that over time, right, essentially we kind of pass all of those mutations through the funnel of natural selection. And some of them, not very many of them, some of them will increase in frequency to the point where they become more common in the population and may increase the fitness of that population. That's the very beginning steps of evolution, essentially. Most mutations don't. They just disappear. They don't get passed on. So if you think about it, if we're, the result of millions of mutations that create us in a sense, think about how many other mutations kind of never got incorporated into our genome. It's kind of cool. All right, so that's it from me today. thank you for all the questions and responses. It was a lot of fun. I'm going to see some of you in lab, the O1 section. Correct. You don't need a lab coat for your first lab, right? It's just going to be safety stuff and some basic statistics type thing. Others, I'll see you in lab tomorrow. What's the day today? Tuesday. Yeah, tomorrow. And if not, I'll see you on Thursday. Thanks very much, everybody.